So, to come to full circle, our last speaker is Chris Snowdon, and he's talking about a role of a very old member of the yeast community, casein kinase, which has been around for many, many years, and he found an unexpected role for casein kinase in glucose sensing. Chris.
Yes, thank you. I want to thank the organizers for giving me this opportunity, of course. And as you mentioned, I want to discuss an unexpected role that I found for these casein kinases in glucose sensing and signaling. So first off, I want to take you through the SRR pathway, something we study in Mark Johnston's lab. So for this pathway, we have several components. In the nucleus, let me find the mouse. In the nucleus down here, we have a co-repressor complex, repressor complex made up of the repressor RGT1, and then the co-repressors MTH1 and STD1. Now, when they're all bound together, when glucose isn't present, they repress Hxt genes. Additionally, we have in the plasma membrane, we have the glucose sensors RGT2 and SNF3. They have long C-terminal cytoplasmic tails that are able to bind the co-repressors as well. Additionally, there's the yeast casein kinases, which are plasma membrane associated, and they're thought to interact with the tail of the sensors as well. Now, when glucose isn't present, as I mentioned, these Hxt genes are repressed. However, when glucose becomes present, the current model states that there's some sort of activation of the yeast casein kinase, and this activation causes the phosphorylation of the co-repressors, and once we get phosphorylation of the co-repressors, they're recognized for ubiquitination, which causes their degradation, and upon their degradation, they no longer interact with the repressor RGT1. We get removal of RGT1 from the promoters of the Hxt genes, and we get expression of hexose transporters. So this is a quick mechanism in order to enable cells to detect external glucose and then produce hexose transporters enabling them to utilize that carbon source. So first off, is the thing that I was looking at was just trying to get an idea of all these components and make sure that they're functioning as we thought they were. So the interpretation was that I would do a few epistasis type experiments and just show that YCK was in fact functioning downstream of the sensors in order to activate the downstream signaling. So the expected result that we were looking at was that if we overexpressed the casein kinases in a sensor knockout mutant, that we would actually see that would rescue the sensor defect in the mutant. So I did some Hxt1 expression assays where you can see in the wild type, when grown in galactose, there's no expression of Hxt1. However, when you add glucose, you get a rapid induction of Hxt1. And as I mentioned in this sensor knockout, missing RGT2 and SNF3, you see no signaling, no Hxt1 expression. However, when you overexpress YCK2, you don't see this rescue that we were expecting in the sensor knockout. So what this suggested to us initially is that there might be some possibility that YCK was actually functioning above the sensor. We double-checked this by looking at another readout, which is MTH1 degradation. As I mentioned, when you have cells in galactose, MTH1 is present. However, when you add glucose, you see this rapid degradation of MTH1. This doesn't occur in the sensor knockout, so we thought that if we overexpressed YCK, we might be able to see some rescue of this sensor knockout phenotype. However, again, we didn't see this rescuing that we were expecting, suggesting again that YCK might be functioning above RGT2 and unable to degrade MTH1. So in order to look at this another way, we kind of just flipped the system and started overexpressing RGT2 in a YCK temperature-sensitive strain. So as you can see, again, in the wild type, we get induction of Hxt1. In the YCK1-2 TS strain, we see very limited induction of Hxt1. However, this time we got the unexpected result of RGT2 overexpression actually rescuing the YCK knockout strain back up to wild type levels and even showing some constitutive signaling under galactose conditions. This suggested and seemed to confirm the possibility that we might actually have YCK functioning more in an upstream role in this pathway as opposed to its suggested downstream role. And this was again confirmed by looking at MTH1 degradation. So in the wild type, we can just faintly see that there's degradation of MTH1. There's such an abundant level of MTH1 in the sensor knockout that's undegraded upon glucose addition. Whereas when we overexpress RGT2, you can clearly see that we get a suppression of that signaling defect. This seemed to be sufficient proof that YCK was definitely having an unexpected role in this pathway. And one of the things that's known about YCK is that it's obviously a kinase, so it's probably involved in a phosphorylation event here. But most importantly, a lot of its study had been done into looking at how it can stabilize membrane proteins, particularly amino acid permeases. So our initial thought was, well, possibly YCK is stabilizing, or is necessary to stabilize RGT2, and this lack of stability in that mutant was causing the sensors to be absent, and therefore you wouldn't have the signaling that we're expecting. So we investigated that, and recently it had been shown that RGT2 was turned over in response to a switch from glucose to galactose. So we investigated that in both the wild type and the YCK knockout mutant, and we found in the wild type abundant levels of RGT2 in glucose, and then upon a switch to galactose, we see the normal degradation after 120 minutes, well, actually after 60 minutes. And then when we looked over here at the YCK2 temperature-sensitive strain, we saw, again, wild-type-like levels of RGT2 and what appeared to be a wild-type-like degradation. So it didn't appear that it was actually involved in any way in the stability of the sensor itself. The one thing we noted, and it's a little more difficult to see here, but it appeared as though the RGT2 species from these YCK-TS cells actually appeared to migrate slightly faster. So that was an immediate clue to us that possibly YCK was directly or indirectly phosphorylating RGT2. So we investigated that by doing some phosphatase assays, where we grew up the cells in glucose and then took a sample and had an untreated and then treated another sample with lambda phosphatase. And what you can see in the wild type is this distinct mobility shift upon treatment with lambda phosphatase, which is absent in these YCK1-2 TS strains. So what we think we discovered is a YCK-dependent phosphorylation of RGT2. So we wanted to investigate that a little further to try to figure out where this phosphorylation was occurring. So just to show you quickly, here's a really busy slide, but what I'm trying to show you is an alignment of SNF3, the other sensor, and RGT2 from several different species of yeast. And what we had identified was two conserved regions in the C-terminal tail. We had box 1, and then just C-terminal to that was this box 2. And what I've highlighted in yellow are what are YCK consensus sites. And what you can see is that they seem to cluster immediately downstream of these conserved boxes. So we wanted to do a few truncations of that tail to see what effect it would have on the phosphorylation state of RGT2. So again, in the wild type, you can see this mobility shift that occurs upon lambda phosphatase treatment. However, in this RGT2 delta box 1 strain, which involves deletion of both box 1 and 2, essentially just taking off all the conserved regions, we see that the mobility shift disappears upon that treatment. Whereas when we just delete box 2 and down, we can see that there still maintains a mobility shift there. So what we were thinking is that the sites for YCK phosphorylation must occur somewhere in box 1 or just downstream of box 1. So again, looking at that sequence alignment, what I've boxed in red here are 4 serines, and in some strains of threonine, that represent the YCK consensus sites immediately C-terminal on box 1. So what we did was mutate those to alanines in order to see what effect that would have on the phosphorylation state of RGT2. And in fact, that turns out to be the key sites in this. So with the simple mutation of just those 4 serines to alanines, you can see that the mobility shift in RGT2 is completely lost, suggesting that this box 1, this conserved region within the C-terminal tail of RGT2 is key for this YCK-dependent phosphorylation. Now the next question was, well, how important is this phosphorylation? We know that in the YCK-TS strains we have almost complete abolishment of signaling, so we would assume that there would be a significant importance to these 4 residues that were involved in the actual phosphorylation event. So we looked at HXT1 signaling in the RGT2 4S to A strain, and we found that in terms of HXT1, it's completely, HXT1 expression is completely gone, and there appears to be no signaling in this strain. As you can see, we express it in the RGT2 SNF3 knockout strain. So when you just have the empty vector down here, you can see there's no signaling. When we have a single copy of RGT2 on plasmid, we get complete induction of HXT1. However, when we express just a single copy of RGT2 4S to A, all signaling is abolished. The same is true when looking at the MTH1 phenotype, where we see that there's turnover when you rescue it with RGT2. However, turnover is completely abolished of MTH1 when you have an RGT2 4S to A strain. So the question became, what is the mechanism by which this is functioning? What's the necessity of RGT2 being phosphorylated in a YCK-dependent manner? And one of the things I mentioned at the beginning was that with this long cytoplasmic tail, we believe that it's important to bring the co-repressors up to the signaling complex in order to get everything to the plasma membrane so that you can have a quick and rapid induction of signaling. So using a split ubiquitin assay, we looked at the interactions between the RGT2 wild type and RGT2 4S to A. And in green, you can just see a positive control and a negative control in purple, just to show you that both systems are working. And then when you look at the red and blue bars, you're looking at MTH1 and STD1 interactions, respectively. And you can see when you're looking at RGT2 wild type, you can see these high levels of interaction between the co-repressors and RGT2. However, just with those 4 point mutations of the serines to alanines within that box 1 of the tail, you can see there's almost complete abolishment of any interaction between the co-repressors and the RGT2 4S to A protein. So it looks as though without YCK-dependent phosphorylation, we're actually not getting the co-repressors brought to the plasma membrane where we think they're necessary in order to be phosphorylated after glucose binding to the sensors themselves. So with that, I'll just take a minute to show you now what the pathway looks like now that we've figured out this new thing. So when glucose isn't around, so let's say in galactose conditions, we have YCK in the plasma membrane. We believe it interacts with the tail itself, but now we know that the interaction is important for actually getting phosphorylation event to happen on that tail. So as I mentioned, right downstream of a conserved box 1, we get a phosphorylation event dependent on these 4 serine residues. And we believe this actually sets up this binding site for the co-repressors and essentially primes the system for signaling. So that allows these co-repressors to come in, bind to the tail. We have a primed signaling event. So then once glucose becomes available to the system, we can bind glucose. And then the mechanism is currently unknown of what's being signaled and causing the phosphorylation of MTH1 and STD1. We always thought it was YCK, but unfortunately now we're on a hunt to try to find some other kinases that might be involved. But either way, you get MTH1 and STD1 phosphorylation, which causes their ubiquitination. They get transported to the proteasome, which they're degraded. And then eventually we get RGT1 leaving the repressors of the HXTs. We get HXT1 expression, and then you can bring glucose in the cell and metabolism can begin. And with that, I'd just like to make some acknowledgements, of course, to Mark Johnson, and then members of his lab, and then Igor and Lucy for some strains and other help with the project. Thank you.
Thank you, Chris. Thank you. Mike.
So I guess 2 questions. One is, it looked like it wasn't just phosphorylation. It looks like you lost an isoform as well. Is that right? Because even in the phosphatase-treated situation, you had 1 band in the mutant and 2 in the wild type, correct? Is that a possibility, or is that not?
Yeah, so when we're looking at the mutant, we don't actually think on that blot that there's a band being lost. I know it kind of seems like phosphatase treatment in the YCK cells that there's a slight drop. I think that's just overloading. I've repeated it other times with kind of uglier blots, but it seems like it's not there. But, I mean, there is the potential that there might be one other.
And my second question is that casein kinase does so many things that I'm surprised that RTG can actually do it. Does that also then suppress the deletion or just the TS?
Sorry, say that again.
Oh, sorry. Does RTG only suppress the double mutant, one of which is a TS allele? Can you also suppress the deletion strain?
Well, the deletion strain of YCK
Yeah.
is not viable. So you have to use the TS strain in order to...
Yeah, well, that's my point. Can RTG suppress a TS?
Oh, yeah, sorry. No, I never thought of something like that. No, we haven't tried to do that type of experiment. But that'd be interesting to see.
There's a question.
Just out of curiosity, is the other sensor completely irrelevant in this?
Well, we're currently looking at the other sensor now. I didn't mention, but the other sensor has a little bit more complexity in its tail. It actually has another box 1 that's even further downstream. And so we've begun to look at it. It seems that it is the epistasis type data still holds true. It is phosphorylated, all those things. But it appears as though it might be phosphorylated by another event as well, that we're just trying to figure that out at this point.
I just wanted to clarify, the phosphorylation of RGT2 is independent of the carbon source?
Yes, absolutely. So it happens in both glucose and galactose. It seems to occur in every condition we've looked at, okay, which would speak to the constitutive nature of YCK.
And while obviously YCK phosphorylates the sensor, why do you say it doesn't phosphorylate the repressor, co-repressor?
Right, so the reason we don't think is because when you overexpress RGT2, when there's essentially no YCK around, you can get effective signaling. So that leads us to believe that if YCK1 and 2, I guess, theoretically could be involved, but there's at least some redundancy that covers what they're doing. So whether it's, one or more kinases that are downstream, there's at least something still missing in there.
So have you actually formally shown that the kinase activity of YCK is required for that? Do you have a kinase dead TS mutant? That, yeah. So it's not a full... Yeah, so this is only the kinase dead mutant?
Yeah.
Okay. And so does this change the ubiquitin status of RGT2? Because I noticed the same thing of RGT2.
I don't know of any ubiquitination of RGT2.
You have not checked this? You
I've
have not
never
tested
looked
it?
at, no,
Okay.
ubiquitination of it. No.
Okay. Thank you, Chris.
Thanks.
Okay.